Hello. It is really quiet in here today. Guess everybody's still waking up after spring break, maybe? Well, thanks for coming. I know there's a lot going on campus, and yeah. So before I get into today's lecture, which will be the last lecture that's going to be on your exam a week from today, I hope you all saw my email with the announcements laying out what's coming up for you. I should also mention that on Thursday, this day after tomorrow, there's a really fun lecture you're going to get on techniques in cell biology that's important for you to know. It's stuff that's not in your book because your book is out of date, but stuff you need to know for your occupations. CRISPR-Cas9 and live imaging, confocal imaging are the two main topics that will be covered. I'm not going to cover them. We're going to do a bit of a guest lecture. So this is Srivatsan Rajan. He's a very talented graduate student in my lab, and he uses those techniques on a regular basis. So it'll be a fun break from me to have him come in and tell you directly about what he does. And the slides are something that, we prepared together, and, it's all sort of nicely put together. So that's what will happen on Thursday. I won't be here, but there'll be somebody who's not quite me, who might look a little bit like me, I don't know, who will be here giving that lecture on Thursday. And that class will be tested on exam 4. That way you have today's class being the last one for exam 3, and you have a solid week to study that material. So yeah, this is Srivatsan, so don't be surprised if you see him on Thursday. So, cool? All right. Well. All right, so a little bit of a recap, especially since we had this long break. Previously on the hit show Cell Biology, we were talking about the different components of your cytoskeleton and every single cell inside of you. Microtubules, actin filaments, and intermediate filaments. We covered microtubules, and very briefly we covered intermediate filaments. So today we're going to focus on actin filaments and what they do inside of your cell. They do a number of things, but one of the major things they do, which is also probably the most exciting thing to look at, is that they are important for cell migration. So we're going to talk about that today, actin filaments and the cell biology of migration. All right. So actins have two major groups, alpha and beta. Alpha, beta, gamma, but they're in two different groups. Muscle-specific ones are alpha actins, and non-muscle-specific ones are beta and gamma actins. For the most part, we'll be talking about beta-gamma today, but some of the things we say will apply to alpha actins as well. Each group interacts with different types of actin-binding proteins and localizes to different parts of the cell. Here is just a really nice image of an example cell. Here's your nucleus. This is your cell cortex. And what's being pointed to here are different sort of sub-examples of the use of actin filaments. So this, for example, this extension out of the cell is a phylopodium, and we'll talk about what that is a little bit later in today's lecture. And then stress fibers, as the name suggests, they help you, the cell, deal with the stress of any movements and so on. So there are different arrangements of these filaments, and that's what's shown here. Remember, with microtubules, by contrast, there was a centrosome, and then you had this almost center point that was the centrosome with this spider web of things that were reaching out from there. And that's not what an actin filament setup looks like inside of a cell. You've got really different actin filaments and different sort of patterns throughout your cell. Okay. Actin filaments are thin and flexible, and you might remember my rope, uncooked spaghetti, and cooked spaghetti analogy for the three different types of filaments we're talking about. Actin filaments would be the cooked spaghetti. They're pretty flexible and wiggly and kind of strong, but not as strong as, let's say, the rope, the intermediate filaments. Okay, so this is what they look like. Actin filaments are thin, somewhat flexible, and have polarity. Remember, we talked about polarity before the break. If two sides of something are different, they have polarity. I have polarity, you have polarity as you look at each other, whereas this thing does not have polarity because the ends look the same, at least up at this point, what I'm showing you. So inside of us, cells have polarity in the sense that their front end and their back end, however that's defined, tends to be different in most cases. And inside of cells, proteins also have polarity. And the most fundamental example of that is the basic protein chemistry you've learned, N-terminal, C-terminal. They're not the same, so a protein has polarity, at least in that N and C-terminal manner. You might recall intermediate filaments don't have polarity because you have N and C terminals for monomers, but when you put them together, you end up with your N terminals on the outside, and they look the same, so the overall intermediate filament doesn't have polarity. Microtubules do have polarity, and then today's topic, actin filaments, also have polarity. So the monomers for actin filaments are called globular actin, globular for globe, so it's round. They're round monomers. You can remember that, G-actin, G for globe, so it must be the monomer. And then the polymer, when it's polymerized, is a filament. So F for filament, F-actin. G-actin is the monomer, F-actin is the filament. This is what it looks like when it's put together. It might remind you of a DNA helix because it is actually a helix. It's a two-stranded helix. The details of how it looks are different from DNA, but the general shape is pretty similar. And so you've got this helix here, which is shown here as a model, and this is an electron micrograph of an actual actin filament. And you can even kind of see on here from the wiggliness of it that it's kind of turning around a little bit. So actin and tubulin polymerize by similar mechanisms, and then I wrote, but also different here. These subtopics are from your book, right? And you will see this as like a subtopic heading. But I want to make sure it doesn't get confusing. Yes, they're similar in some ways, they're different in some ways. I'm going to tell you about that today. And I'll try to point out the similarities and the differences as we go. So the first place we see similarities and differences is when we look at how they're formed and how they're pulled apart. Ignore this for a second, and I'll remind you of this. This is microtubules, not actin filaments, what we talked about before the break. Right? If you might recall, I told you about dynamic instability. And my analogy is if Chicago could build out roads and highways however it wanted, whenever it wanted, build it out, take it apart. Build it here, take it apart. That's what your cell does with dynamic instability with microtubules. It's able to quickly assemble and disassemble these microtubules to create highways to make things go where it wants it to go. To that end, it uses the system we talked about where your plus end, the growth end of the microtubule, has a GTP cap that stabilizes it. That GTP is bound to the one of the two tubulin subunits. And as time passes and you lose that cap, there's a vulnerability for these hydrolyzed, for these subunits that had the GTP hydrolyzed to GDP, shown in light green now, that once that becomes available to the outside of the tip, it starts to come apart really quickly. There's a video I showed you where it quickly disassembles, and that results in what's called catastrophic shrinkage. And now you can then put a cap on and start building it back up again and back and forth. And it sounds random and chaotic because it is, but in reality it's carefully regulated by things that aren't shown on this particular slide, but that we briefly talked about. Things like microtubule capping proteins or stabilizing proteins that will actually help this cap stay in place and protect it from being degraded. So there's a balance that the cell maintains based on this relatively chaotic system. That was microtubules. By contrast, actin filaments do something called treadmilling. And the name treadmilling is very appropriate. It is like moving on a treadmill. If you imagine somebody walking along here, they might not move, but the actin filament will change. Because what's happening is that you are adding monomers at the plus end, just like you're doing here, broadly speaking. You're adding monomers to make the filament longer. But instead of taking things off of this side, right, that's what microtubules were doing, you don't do that here. You only add on this side, and you take them off from the backside, the minus end. So with microtubules, that wouldn't work because the minus end is anchored onto the centrosome and is nice and protected, right? That's just where things start from. With a microtubule, everything's happening at the plus end. Take it off, put it on. But with actin filaments, it's different. You put it on at the plus end, but you're removing the monomers at the minus end. So if you look at this diagram, as time passes, in the case of the diagram, they just lined everything up nicely here. But really, if you keep adding more and more here, they should have drawn this with sort of like a, they should have drawn like this one should have stayed here, and if they'd drawn one here and then drawn one here, you'd see that it's getting longer in this direction because you're adding, and then as these come off, this part's getting shorter. So without the filament, while this part of it stays the same, you take stuff off from one side and you add them on the other side, and the movement treadmills. The actin filament will actually, as a whole, end up in a different location because you're taking stuff off from the back and putting it on the front. Kind of like how a treadmill works, if you think about it. So that's why it's called treadmilling. All right? So there's some similarities and differences here. You are adding monomers to make a filament. You're taking off monomers to reduce the size of your filament. But with microtubules, it's all happening at the plus end with this GTP cap, GTP tubulin. With actin filaments, it's happening, the additions at the plus end, the removals at the minus end, and I haven't yet told you what drives this process. That's coming up next. It's not GTP, so it's another difference. GTP works with microtubules. Actin filaments use ATP. And you can remember this because actin starts with an A and ATP starts with an A. So actin for ATP and then microtubules with GTP. Yes? Microtubules. Yeah. So dynamic instability involves microtubules, and then treadmilling involves actin filaments. Yeah. Okay. So this is just a more detailed diagram of what I've kind of already told you, and these two things show you the same thing. This one just has more detail, so I put both on here so that, you know, whichever one you like, or look at both, and it helps you understand it. It's what I just told you here is shown in more detail. So here at the plus end, you are adding on these globular G-actin monomers. And then as they're added with ATP attached to them, similar to how these are added with GTP attached. But again, instead of GTP, you're using ATP. So you add these here, they have ATP attached, and at some point after the addition, they're hydrolyzed. So they get turned into an ADP attachment, as shown here, ATP all along, and then this one here is hydrolyzed, and you have ADP. So again, this is really similar to what happens here. You had the GTP, it's going to get hydrolyzed, and you have GDP, but you're just doing it with a different nucleotide-based molecule. As that happens, the hydrolysis decreases the stability of the polymer. In other words, this part of it is much more stable now than this part is because it's got the ADPs instead. And so at some point, the unstable ones at the very end here are more vulnerable to being popped off, and they pop right off and come off. Now, once it comes off, you've got more monomers that are sitting there, and the cell could come in and add another phosphate group. It takes energy to do that. Now you can go ahead and reuse those at the plus end. In this case, remember the cell is moving, right? That's the main thing I'm telling you about. This is actually a really important balance because if you want to move quickly, you better be able to have lots of monomers to add onto the front end, the plus end of your actin filament. Otherwise, how could you grow quickly? Inside of a cell, as you'll see, you're adding hundreds and hundreds of these filaments rapidly to propel your movement forward. So it makes sense to do that. You have to have a lot of raw material. That raw material is these monomers here. So as they come off the backside, they end up being phosphorylated pretty quickly and then reused for the front side again. The same thing is shown here, just in a little bit more simplified diagram. Actin with bound ATP is being added, and then actin with the ADP bound is being removed, and then here's your addition of your phosphate group. Any questions about this? Yeah?
So is there a change in angle, which was in microtubules, when the GTP and GDP tubulin comes out? So is there change in angle?
You mean at the site of the binding?
Yeah.
Yeah, so the question is there a change in the angle? Is the site of binding of the ATP or the ADP changing the conformation of the monomer to the polymer? Yes, yeah. So all of these things kind of come back to shape changes, that you are modifying something that couldn't previously bind into the polymer, and now with the ATP being bound, it's able to slip right in. So yes, good question. Yeah? Yeah, so the question is about, good point. A question is about, okay, so you've released these guys here. Are they the ones being phosphorylated and being put back in here, or are there different ones that are going in, different filaments? Right. So this diagram is super simplified. In reality, this would be like a bunch of them, and they would all be releasing a bunch of monomers from their backsides, their minus ends, and then being phosphorylated, and then whatever was most convenient to be grabbed to put on the front end of a new actin filament would be grabbed. So it's not necessarily the same ones, but it could be. Yeah. Okay. So the function of actin filaments then, based on what I've shown you, a little bit of a summary, depends on the dynamic equilibrium between filaments and monomers. And actin binding proteins stabilize actin filaments. Remember, again, before the break, I told you about microtubule capping proteins, microtubule binding proteins. Similar concept. The actin filaments, when they have this ATP bound monomer added, can be stabilized if you are using different proteins that we're not going to get into in this class to help stabilize those filaments and keep them in place. Okay. All right, now I'm going to show you just a quick summary slide from your book to illustrate the diversity of the proteins that bind to actin and modify it. I don't expect you to memorize this at all, but you will hear about a couple of these proteins later on. So these are just some of the examples of what actin filaments do and how they are interacting with other proteins. Most of these are things I wanted to just give you a sense of, because if you ever see them in an entrance exam or something, you actually don't really need to have it memorized, because the name often tells you what it does. So a severing protein, for example, if you know that severing something means cutting it, then you can pretty correctly guess that a severing protein is going to chop up an actin filament, right? A bundling protein. What's a bundle? You put a bunch of things together. So bundling protein is probably bundling actin filaments together, and that's exactly what you see here. So you get the idea. A lot of these proteins are named, thankfully, in a very self-explanatory way. A capping protein, right? It caps the plus end. So stuff like that. We will today talk about myosin motor proteins and about nucleating proteins in a little bit more detail. All right. So the next sort of topic is that a cortex rich in actin filaments underlies the plasma membrane of most eukaryotic cells, and then cell crawling depends on that cortical actin. So I've been telling you that you have to make these filaments to move, right? But if I'm a cell and I'm this big and I've got actin filaments in my very center that are being formed, you can imagine that's not really going to help you move as much. You're in the middle of the cell. You need to have actin on near the boundaries of your cell, because your boundaries of the cell are what move, right? So we'll see a diagram of how that's put together. Imagine thousands and thousands of actin filaments. That's what this orange represents. The actin cortex is basically a high concentration of actin filaments that are near the surface of your cell. This is a lamellipodium, which we're going to talk about a little bit more in detail later on. What a lamellipodium is a flat, fan-like extension of the cell. So if you remember those toys that you can throw and they stick to something, I think I used this analogy once before, those are kind of what your lamellipodia look like, and they're extensions of the cell. And we'll come back to that in a second. Substratum is just a fancy word for saying what is a cell on top of, the substrate that it's on top of. So if you see that, that's what that is. So this cortex is pretty much always under tension. If you're moving as a cell, you're going to have forces pulling you in different directions, and it needs to be able to withstand and that tension. Actin polymerization at the plus end protrudes this lamellipodium. So this structure is going to keep moving out, because actin filaments here have more actin monomers being added to the plus ends, which then extends this cortex out. So that's the first step of cell migration, is that you have to push out in the direction you want to go. The second step of cell migration is, you'll notice in this diagram there's a little bit of a white space between this protrusion and the substrate. So cells have to stick to something when they migrate. I think sometimes when we see videos and you see a cell moving from point A to point B, it looks like it's just kind of flying through the ether, but it's really not. Cells have to bind to something to move, just like we have to be on here to walk. All right? If I tried to walk up in the air, it wouldn't work so well. And so these cells have to be able to bind to a substrate to move. So after they extend out that first, okay, I'm going to go and see what's out there, then it has to slap down on something. To slap down on something, it needs to be sticky in some way. And the way it's sticky is it uses these things called focal contacts. So they're focal contacts, focus, or is like a small area you're focusing on something, and then you're contacting. So your focal contact is like a really tight contact it's making with the surface. And that attachment now lets you hold your extension in place. These focal contacts contain, among other proteins, a protein type called integrins that are really important for this binding to happen. So that's the second step, is you have to stick. Once you've stuck, the third step involves the backside of the cell. So you'll notice there are these little red dash lines drawn here, here, here. These are focal contacts that already existed, because of course, before I can walk, I have to be standing. So I'm already stuck to the floor. Well, how do I get those off? If they were sticky, how do you make them unsticky? One of the things that a cell does is it contracts the back of its cell. And we'll talk about how that happens, but I'll tell you right now, it uses actin and myosin, because myosin has a contractility to it that complex does, and you basically pull the cell off of the surface. Right? So it'd be kind of like if I was walking, and I'm like, oh, I'm stuck back there, and I turned around and I just grabbed my backside or something, and I yanked it off and kept going. That's kind of what your cell is doing. It moves forward, slaps down, then pulls its backside up, and then repeats the whole process over and over again. So advance, push out, stick to a surface or adhere, and then contract your backside. All right, so let's look at these. Yes?
So it's kind of like a slug motion where you're just kind of using the same regions to propel forward rather than like a circular?
Yeah, so the question is it more like how a slug moves, where you're using the same things over and over as opposed to propelling? Yeah, it's a great analogy. In fact, you'll see lots of, without going too off topic, examples in biology where some of the macro level movements and behaviors mirror things at the micro level too. So yeah, absolutely. If it helps think of it as a slug moving, that's a good way to think about it. Yeah. So cell motility then, as you gathered, dependent on different actin filament structures. These are two that I briefly mentioned that this is a more clear-cut example of. At the front end of the cell, you have phylopodium, that's the singular. Phylopodia would be plural, and lamellipodium, which would be lamellipodia as plural. So lamellipodia and phylopodia, they're pretty similar. They're extensions of actin, but they look very different. Lamellipodia are those flat fan-like structures I mentioned. They're bigger. They kind of just slap out there. And then phylopodia tend to extend out as sharp, pointy little things that quickly extend, retract, extend, retract. And you could think of these maybe as, the lamellipodia might be sort of, so the cell needs to know where it's going, right? So maybe you can think of the lamellipodium as like a squad in the army that's out on patrol and looking to see what's happening. So the lamellipodium is like looking out, okay, what's going on over here? Let's go over here. What's going on over here? And then the phylopodium is sort of like the point person on that patrol, because the phylopodium will stick out even further and send something and come back, and stick out this way and send something and come back. So they work sort of in concert. The phylopodium is more transient and sharp, and the lamellipodium really slaps down and makes an imprint and then helps the cell move forward. Both are composed predominantly of actin. All right, so how is all this happening? You are inside of a cell. You want to make these structures and move forward. You want to make new actin filaments. How do you know where to make them and in what direction? So the nucleation or formation of new actin filaments requires something called the ARP complex, which moves the plasma membrane forward. So this is just a nice picture from your book of a dense network of actin filaments. And this is how these networks are formed. So if you were starting back here and you have this in pink actin filament going this direction, and this blue thingy here represents a cap, all right? So it's stabilizing this side of the actin filament. And so this would be the minus end, and you can see these monomers that are coming off of it. And the cell wants to move this direction. What it does is it has these ARP complexes, shown in this brownish color here, bind at various points of existing actin filaments, and they bind at an angle. It's not exactly, it's not like you can keep going in the same direction, but because of the way these complexes are shaped, they'll bind and they'll send out a new actin filament, they'll start a new actin filament in an angled direction. And then if you send out a new one in this direction and then say a new one in this direction, it's not very efficient, but because you're not going straight in the direction you want to go to, but eventually you end up creating a network that goes in the direction you want to go. And actually it ends up being a very strong network because of this interlacing, right? So that's one advantage of doing it this inefficient way, is that you have this strength to it because you're crisscrossing all the time, but your crisscrossing is directed in the direction you want to go. All right, so ARP complexes are nucleation sites for new actin filaments. The analogy for microtubules would be what? What was your nucleation site for microtubules? The centrioles, or the centrosome, which has centrioles in it. So whereas with microtubules you had that one spot, the centrosome, with centrioles in it, that was sending out new microtubules, here you don't have the one spot. You actually make lots of new sites of nucleation of actin filaments wherever you need to as you go. So the general concept is the same, but the specifics are very different. All right, actin associates with myosin to form contractile structures, which the example I've given you so far is pulling that cell's backside up. The back of the cell needs to come up. This goes back to motor proteins, which you heard about with microtubules. Microtubules, you may recall, work with two motor proteins, dynein and kinesin. Dynein moves towards one direction and kinesin moves towards a different direction. Does anyone remember which direction dynein goes in? Minus. DM, direct message, or MD, medical doctor. DM, MD. All right, dynein goes towards the minus end. Kinesin then goes towards the plus end. That's microtubules. This is similar to that. Myosin is another motor protein, so now it's a third motor protein, and it moves usually, as shown here, towards the plus end. I'll tell you right now that for this class we have to simplify this. In reality, you have roughly 40 different myosin genes in your body that are put into 2 dozen or so subclasses of myosin that do different things, moving in the minus, the plus, or whatever. for this class, we can't get into that level of detail. So for this class, what I'm teaching you is that this myosin, as shown here, moves towards the plus end. All right, and that is the most common direction for myosins to move in. So here's your myosin. Remember, dynein and kinesin, they walk kind of like people do almost, as we were talking about in office hours today. And, speaking of office hours, don't forget, I have extended office hours after class for an hour and 45 minutes from 12:15 when class ends to 2 p.m. upstairs. All right, because again, exam's coming up and because I can't do office hours on this Thursday. So please come see me if you have questions about anything at all. Anyway, okay, back to this. So myosin then moves towards the plus end. Shown here in red is an actin filament. This is that double helix shape that I've been telling you about. Here's the plus end of it, here's the minus end of it. In the same way that microtubules can be transport mechanisms for organelles and vesicles to move the highways of the cell, actin filaments can do that sometimes too, and that's what's shown here in this panel B, that you can have a vesicle bound to myosin walking along actin filaments, just like what we learned about with microtubules. But what we'll focus on today is a different thing that myosin can do. When it's bound to something where it's not able to move, its walking movement will move the actin filament. So it can actually move the actin filament as the myosin tries to move to the plus end in this case. It can't because it's stuck on this membrane. That walking motion is going to actually end up kind of like moving a rope or something. It's going to move the actin filament in this direction. All right? That might remind you of what happens with microtubules inside of a cilium or flagellum, where the motor protein is stuck in place, but it's able to bend the microtubules because it's stuck in place. So that was another example we learned last week about how motor proteins, when they're stuck to something, can transfer their energy to moving something else. Okay. We'll come back to this myosin in an example in a bit. Extracellular signals can alter the arrangements of actin filaments. That's probably not a surprising statement. We've been talking so much about cell signaling in multicellular organisms. Everything depends on everything else, GPCRs, RTKs, blah, blah, blah. So it makes sense that if you added signals to cells, they would respond to it and behave differently. These are just some examples that are remarkable to see. This is the same cell, or at least probably representative examples. This is the cell unstimulated, and all the stuff inside is actin. Then somebody added a molecule called Rho, which we're going to learn about in a bit. And look at how different the cell looks now. They added something called RAC, see how different it looks, and they added something else called CDC42, and see how different it looks. We're going to learn about Rho and RAC in the next few slides. They're really important for migration. CDC42 you won't hear about until we get to a lecture about cell cycle stuff in a few weeks. But just an example of how different signals can change actin inside of cells. All right, so to summarize what we've learned so far, 3 different major components of the cytoskeleton. This is the summary slide and another one I gave you that I suggest using to help organize these thoughts. And I've shown you some examples of all the migration that can be dependent upon this type of a movement, right? These are just some of the things I've previously shown you. Cells migrating, they look so pretty, what are they doing? And now you can imagine inside of here, these little extensions, might be lamellipodia or filopodia. You can imagine inside here actin helping these cells move. So you're starting to get a sense of what's happening inside of these pretty images and videos that I've been showing you. And here's just a gorgeous image of actin and microtubules together. So here's your cell nucleus, microtubules, coming out in a relatively straight pattern because you have your centrosome near your nucleus. You might have a couple of centrosomes here. I'm guessing it's right about there based on the patterns. And then meanwhile, look at your actin. It's so different, right? You've got this cortex I told you about at the boundary of the cell, and then you've got a bunch of other patterns of actin everywhere else. And look, here you can see lamellipodia or maybe even filopodia coming out. So all of it kind of put together. In the next several slides, what we're going to do is talk in detail about migration. Right? I've given you hints of that. Lamellipodia, bind to a surface, pull your back off, all that kind of stuff. And now we're going to put it together a little bit. The next few slides also have a ton of text on them. You may have noticed I try not to do that. I don't like to read from slides. I don't like you to have to read a bunch of stuff, but sometimes it's useful. And because this stuff is so complicated, I'm going to give you slides that have a lot of background information, and I've bolded the things that are most important for you to learn. So if you really don't want to study hard, I guess, you could just read what's in bold. But to study better and to learn, make sure you read the context to help you understand what's in bold, all right? The first thing we're going to talk about then is polarization. And I already told you about polarization. Cells have polarity. Migrating cells have to have polarity. So cell polarization, in bold here, refers to the tendency of migrating cells to have a distinct, stable front and rear. That's all it is. Your front's different from your rear. This information gives you a lot of stuff if you're interested in cell migration, about how that polarity comes up and how it's maintained. Again, just to be clear, I'm not going to test you on all this stuff. It's there as background information to help you understand the important points that I am asking you to learn and that I will test you on. So let's talk about an important major point then, the leading edge of the cell. I've already told you about the leading edge of the cell. So these slides will be a bit redundant on purpose. I'm putting together information I've already given you. The leading edge then has this intense actin polymerization that generates a protrusive structure and adheres to a substratum. The trailing edge, the backside, has stable bundles of actin and releases and dissembles adhesions. This is all saying what I've already told you. The front of the cell, the leading edge, is making new actin and generating protrusive structures, lamellipodia, filopodia, and binding to different things. Meanwhile, the backside is not doing that. They're stable bundles of actin, and it is instead pulling up those adhesions to let the cell move forward. This video, which is kind of nice to get a sense of this, has a GTPase called RAC. The warmer colors mean there's more of it. The cooler colors, the blue and green, mean there's less of it. And the cell is going to move this way. And what I want you to notice is that there's a lot more RAC protein at this front end, shown by the red, orange, yellow. And on the back side of the cell, there's very little RAC protein. That's an example of molecular polarity, or protein polarity in a different sense. Sorry, I shouldn't have used that term. That's an example of the polarity of a protein inside of a cell, where you've got a lot more of it on this side than this side. So, as I'm going to show you in a second, RAC, as you might already guess, is really important for the leading edge of the cell to migrate, for that activity that we've been talking about, the formation of actin filaments, and we'll come back to that. All right. First things first, I want you to ignore these panels here. I may just take them out in the future. I don't want to get into that level of detail. But I do want you to focus here and to not think, oh my God, I have to know all these things. Instead, I'm going to point to a couple of things for you. This picture here of a cell really integrates a number of concepts you've already learned about. A chemokine, you don't have to know what a chemokine is. You can see these dots here binding to this here and get a sense. What is, generally speaking, this type of a molecule called? A ligand, right, or a ligand, however you'd like to pronounce it. And this is a type of receptor. Based on the shape, what kind of receptor do you think this is? GPCR, right. So GPCRs are 7 transmembrane protein receptors, 1, 2, 3, 4, 5, 6, 7. And when you start studying, you'll remember that this is the shape of a GPCR. As it turns out, chemokines are just FYI, molecules that tell a cell, move towards me. So if I secreted a bunch of chemokines, cells would want to move towards me. That's what the signal from a chemokine ligand says to do. So inside of a cell, this is some of what happens. You end up polymerizing actin on the side of the cell that's getting that signal so that the cell wants to go, because the cell wants to go this way, so it needs to have actin being polymerized to go that way. Here's that RAC protein that I showed you right here. Turns out that RAC is really important for stimulating actin polymerization. And you can remember this because, the way I remember it is RAC active, RAC for activity, active, move forward, so that's how I remember it, if that helps. And then on the backside of the cell, we haven't talked yet about the contraction in detail, but you can see what I've already kind of mentioned to you, that there's actin and myosin. Myosin is shown here, I guess, in blue, that is going to be responsible for pulling the backside of the cell up. All right? And this molecule, Rho here, I may have mentioned to you, I'll talk about it in a few slides, is involved in this process. So Rho is also GTPase. And so when I remember RAC and Rho, yeah, I showed you them on that slide with the, here you go, Rho and RAC. So RAC activates actin polymerization, and you can kind of see that you form these nice new growths of actin. Rho does kind of the opposite. You have stable actin filaments that aren't doing much of anything in terms of growth, and you end up looking something like this. So when you're talking about migration, RAC activity, and I think of Rho as Rho is slow. That's how I remember it. So the backside of the cell has a lot of Rho. It's slow. It doesn't want to go that way. and then the rack on the front side is going to help make new actin, make the cell go this way. All right, and we'll come back to that in a bit. Protrusion. We already talked about protrusion. So here are some beautiful pictures of the two protrusions I was telling you about. Lamellipodium, this is a lamellipodium. You can see it's flat, fan-like. And then these spiky things are filopodia. And if you look carefully, you'll notice that the filopodia are coming out from what looks like a lamellipodium. And they are. So again, you have your scouting party, and then you have a pilot. You have like a front person who's going out there and getting more information and bringing it back in. So protrusion is the de novo, or brand new formation of membrane extensions or protrusions in the direction of migration, the leading edge. These are things we've already talked about. The plasma membrane has to expand if you're going to go anywhere. You have an underlying backbone that's composed mostly of actin, and then you have contacts that have to be made with the surface that they're migrating on. This we already talked about. It's produced by local polymerization. This is exactly what I said, so I won't repeat it. Lamellipodium, filopodium, we've talked about them. And then this is just expanding on what I told you. The filopodia, the spiky ones, act as sensory devices. They're kind of looking around to see what's out there, whereas these guys are providing the surfaces for the movement. We also talked already about ARP23, and here it is again. So ARP23, remember the ARP complex, attaches itself to the side of a pre-existing filament and gets new actin to polymerize at a fixed angle. That image I showed you of the angled formation of new actin filaments. Monomers are provided because you chop off the backside of older filaments and you make the new monomers out of them, and then you have these two proteins that are very important for this. Cofilin is an actin severing protein, and formin is a protein that promotes actin formation. And you can remember this because formin has the word form in it. So you're forming actin filaments with formin. If you remember that, then you'll probably remember that the other major protein, cofilin, does the opposite. Okay. Yes? How do the filopodia sense? That's a great question. The short answer for the purposes of this class is that this right here, while in this diagram it's shown as on the cell membrane, imagine if you instead had a spike coming out here and you had that receptor at the end of the spike. That's actually what happens.
That's what I thought.
Yeah. Yeah.
But so like the filopodia don't interact with
Any outside stuff, like yeah. The question is, do the lamellipodia similarly receive signals from the outside that they're sensing? Again, it's sort of like it depends is the easy answer. To my knowledge, lamellipodia that aren't filopodia don't do a lot of sensing, but I'd be shocked if they don't do some sensing. And of course, to bind to a surface, you have to do a whole other type of sensing. So you're certainly doing that. Whether or not they're also responding directly to a signal saying move towards me or don't move towards me, I'm not sure. I suspect there's less of that. But even though I talk about these as separate structures, in reality, you have this lamellipodia with filopodia coming out. And you can imagine that the cell's expression of a receptor to sense something may not be so carefully confined just to the filopodium, right? It might actually be extending somewhat down from there. So, yes?
Does this lamellipodia attaches to the surface through the tethering mapping?
Okay, so the question is, how do they attach to the cell surface?
Does it occur through tethering?
Through tethering. It occurs through, we're going to talk about actually in one of the slides here. It occurs through a number of proteins, including a protein family called integrins. There are sticky proteins and cadherins that extend out from the cell and will bind to stuff. We'll get to that in a couple of slides. Yeah. Okay. All right. This is a really nice, fun example of this. So here's a lamellipodium, and then you have filopodia coming out. And notice how they form and then they almost fold back on themselves. This is all actin and white. And you can see the time scale. I'm going to play this again before I play this one. Notice how quickly this is happening. This is in seconds. So you have really rapid formation. And you can see here these little sparkles almost where you're forming new filaments or taking apart old filaments. So it's a really active process. This one, I believe, doesn't have any audio, but it just shows you this as a model. So you have a cell here. You have a lamellipodium forming, what you're seeing here, and then the retraction is going to happen on the backside of the cell. And I might speed this up. It's a little bit slow, but basically, here we go. This is the part of the video that's actually important. So if you zoom into the front side of the cell, this is a video of what I've been telling you about. This is the formation of new actin filaments, right? So here's an ARP2/3 complex in red. Here you have actin monomers binding, and as this filament grows, it pushes the plasma membrane out in this direction. I think this doesn't have any audio. Yeah, it doesn't. And then going back to that chemokine and its receptor, now it's actually shown a little bit more accurately. If this is a lamellipodium or a phylopodium, it doesn't really matter. You have a signal coming in, and now that signal promotes the ARP2/3 complex to form new filaments to move the cell in this direction. So, all right. So that kind of gives you a sense of it, and then this diagram down here summarizes all of that. It's something I've already shown you, that you're going in this direction, up, in this case up this way, and you're making these new filaments here based on these ARP2/3 complexes. Here's cofilin in this very light gray back here, and it's helping to chop up the old actin filaments. And then while it's not shown here, formin would be more prevalent here for helping to form and maintain the new filaments. All right. A little bit more detail about ARP2/3 to see it a little bit better. This is just a simple diagram. So here's your ARP complex. It's not doing anything. You have an activating factor coming in. Very vague term, I know. This is just to kind of illustrate that you form this complex when it's active. It takes actin monomers and it makes new actin filaments. So there's not a ton of new information on this slide. It's just to reinforce what you've already been told. This is a nice example. This is in seconds over here, where somebody put plastic beads in a dish, coated it with these activating proteins, and added in actin monomers and an energy source, and then watch how quickly things will just form actin filaments off of this bead, just onto the bead, based on what you put in there. So within like 10 minutes or less, you end up with this nice network. Okay. Any questions about any of this so far that I haven't answered? Yes? The question is the rack directly activating the ARP, or is it activating something else that's activating the ARP? So it is part of a pathway. Let's just go back to it, because I don't actually honestly remember exactly what it looks like. Here you go. So yeah, it's not shown here. I'd have to go back and check if it's direct, and I want to say it is. I think that rack directly stimulates ARP2/3, but it's possible there's something in between as a step. I don't remember that. Good question. Yeah. Any other questions? Okay. All right. So this is just, I'm not going to spend a lot of time on this one. It's just a cool, fun, well, not fun for the person who has this, but it's a cool, fun example of how bacterial infections can co-opt this mechanism. So listeriosis is a common bacterial infection that has a lot of really pleasant symptoms. You can get it as basically food poisoning. A lot of times you might have a listeria infection, and it can be quite serious, particularly if you have if you're immunocompromised or if you're older. So it can be quite bad. But in the U.S., as you can see, it's pretty rare to actually die from this. One of this bacterium, the way it works is it gets inside of a cell, and it uses the cell's network to make itself a tail made out of actin filaments. And then once it makes enough of those, it pops right out of the cell and goes to the next cell. So it's infecting cells, and then it's using the actin assembly machinery to make itself a tail so it can swim around and go to the next cell. And this is a nice video showing that. And actually, I think this one does have audio, so let me make sure that's up and running. Okay.
This mammalian cell has been infected with pathogenic Listeria monocytogenes. These bacteria move throughout the cytosol by recruiting host cell actin, which polymerizes and pushes them forward, producing a comet's tail in their wake. Whenever a bacterium is pushed into the plasma membrane, it creates a temporary protrusion and is then bounced back to continue its random path. If we look closely, we can see a bacterium divide inside the host cell. Immediately after separation, the two daughter cells assemble their own actin tails and start moving about. These bacteria can also form actin comet tails and move in cell extracts. Here, the bacteria are expressing the green fluorescent protein, and actin is labeled red with a fluorescent dye. The dynamics of the actin tails that propel the bacteria through the cytosol can be modeled based on known biochemical and physical properties of actin and actin filaments.
So it's kind of neat when you think about this is, if you think about evolution and sort of a battle between a host and an infection, a bacterium might say, I need to evolve the ability to move so that I can do what I need to do to infect. In this case, the evolution was, well, I could evolve my own ability to make an actin tail, but what if I just infect and use the cell's own machinery to do it? And this is similar to how viruses will infect you and use the cell's machinery as well. So there's a lot of interesting things there to kind of learn about if you're ever interested about how different evolutionary battles happen in a sense in terms of trying to survive, whether it's the infector or the host. Okay, so then the next aspect of this, which you asked about, is adhesion. How are you sticking to the cell? So I told you about focal adhesions as zoomed in here. And if you zoomed into this focal adhesion, you'd see a bunch of stuff. So basically, this is the cell membrane, these lines right here. This is the extracellular matrix, all the goop outside of the cell that it's moving through. You can think of it as a network that it has to make its way through. And then this is a complex of proteins that helps it actually create these focal adhesions. And we're not going to talk about the vast majority of these. But one protein that's really important in terms of its actual end result is integrin. Integrin is a protein that crosses this membrane and is able to stick to and bind to this ECM, and that's how it forms those adhesions. So if we look at the parts that are bolded up here, adhesion to the substratum occurs mainly via integrin receptors. And this is some detail about what integrins are and how they work. Basically, they link the substratum, the outside of the cell, to the actin cytoskeleton, the inside of the cell, and provide the traction for migration. Sites of adhesion are usually restricted, and they vary from being small ones, nascent or new adhesions or focal complexes, to large and elongated focal adhesions. So this is a terminology difference, a focal complex versus a focal adhesion. I'm not going to test you on that kind of a difference. I don't really care if you want to call it a complex or an adhesion. But if you see these things, that's why they're named that way. They are different. So it's a good idea to learn something like that. Focal complexes, focal adhesions, their purpose is the same. They're just the size is different. All right. There's a lot of different molecules that are part of this. There are signaling complexes that contain kinases and adapter proteins. You've already learned about those general types of proteins. Paxillin and FAC are two very important signaling components. They're both implicated in regulating the Rho family of GTPases. And remember, Rho helps the cell go slow, so this kind of fits together now. Rho, if you're not moving over there, you're sticking, right? If you're sticking, you're not moving. If you're not sticking, you're moving. So it makes sense in a general sense that the Rho family is involved in helping these sticky things, these focal adhesion complexes form. And then this complex in turn regulates the polymerization of actin, actomycin contraction, and microtubule dynamics. And we're going to, in the next slide, or next couple of slides, we'll look at more detail at actomycin contraction, because that's the one thing we really haven't covered yet. This is an example of that. What you're going to see in this is paxillin, one of the proteins I just briefly mentioned to you. That's an important component of this adhesion complex. So here's paxillin in green. So in this video, it's tagged, and you can see how that paxillin is so prevalent on the front edges of these cells, because remember, those front edges, the lamellipodia, are reaching out and going splat to hang on to something. So it makes sense that they would have to express a lot of this paxillin as part of that complex to do that. And I think this is just more of that, kind of showing you different examples of it. Okay, so the last thing we'll talk about as part of this series of this subset of the slides is the regulation and integration of all of this. I've now kind of shown you migration in two different ways. First, at the very beginning of class, I introduced how actin forms and what it's doing and the filaments. And now I'm giving you more of a process-based overview, that you have to reach out, you have to stick to something, you have to then make actin to move yourself forward, you have to pull your backside up, which we're going to talk about. And so you can think of it as processes and the elements of those processes and try to integrate that together to get a sense of cell migration. So to summarize a little bit of what we've talked about, it's regulated by complex signaling networks. Okay, no surprise there. Integrins are an important part of the binding. Local transient signals retain polarity and drive processes such as things we've talked about, the polymerization of actin, the adhesion, the actomycin bundling and contraction, which I'll tell you about in a second, and microtubule dynamics. So large signaling scaffolds, this is just more information if you want it, are at the heart of this regulation, which is a fancy way of saying lots and lots of proteins get together to do this. And again, here's a complex diagram of that. where you have cell moving in this direction. Now what this diagram is doing is integrating the extracellular matrix into the diagram. So here's the outside of the cell, and here are integrins and another protein type that we are not talking about today that together are actually anchoring the cell to this surface. And if you look in here, here's that RAC kinase I told you about. Here's the Rho, or the RAC GTPase, and here's the Rho GTPase. There's paxillin. So you've seen some of these things already and how they're all put together. Obviously, anything I'm not emphasizing, I'm not going to test you on all of this. But again, just pay attention to the high points and use this as learning material to help you understand the process. So, yeah, I said I'd talk about actomyosin next, but one more slide, then I'll do that. RAC and Rho. I've been telling you about them. This slide just summarizes RAC and Rho in one slide for you. So remember, RAC for activity, or basically moving the cell forward. Rho makes you go slow, contraction, not really moving the cell forward. So these are some of the things that these different GTPases do. They are maintaining a balance. As written here, RAC activity generally promotes motility, whereas Rho activity generally promotes stability of the cell. As you've already heard me mention, RAC and Rho are small GTPases. You've already learned about GTPases many, many times. GTPases are proteins that will turn GTP into GDP. And so they act like switches in cell signaling pathways. You've heard about that too in previous lectures. So you may recognize this diagram, the molecular switch diagram, just with Rho put in instead of something else. So in this example here then, you've got Rho, you have GTPase activating proteins. Remember those from when we talked about signaling? GTPase activating proteins, GAPs, will activate a GTPase. So they're helping to activate Rho, which is a GTPase. And then that encourages Rho to undertake its activity, which then gets rid of which then changes GTP to GDP. Then on the other side of it, you've got GEFs. Remember, guanine exchange factors. They help promote the exchange of the nucleotide on this side, where you take out the GDP and you put in the GTP instead. This is the same thing you've learned about a few weeks back, just applied now to the concept of cell migration. All right, so the last on this topic slide that I want to cover is what I keep saying I'm going to talk about, the retraction of the cell. It just made sense to talk about it at the very end. So it's propelled by a coordinated contraction of the actomyosin cytoskeleton. And as this says, it's actually not well understood. So I'm not going to give you a ton of detail about it. If you look at this diagram here, here's the front part of the cell that we've really talked a lot about, filopodia, actin, lamellipodium, et cetera, et cetera. On the back side of the cell, you need to pull this off. And so this would be a focal adhesion complex where it's stuck to the substrate below it. In green, you have these actin filaments. In red, you have sites of adhesion. And what's actually happening here is that you have actomyosin complexes where, remember I told you that myosin binds to actin as a motor protein, and it could either move stuff along actin or it could make the actin move. So in this case, if you've got myosin stuck somewhere on the plasma membrane or close to it, and you have it binding to a bunch of actin back here, it can kind of grab the actin like it's a rope or something and just yank it and pull it basically this way. And as it does that, all the stuff here, this adhesion site that the actin is attached to, is yanked off of the surface of the cell. It's very much a physical process. You've got a sticky bunch of proteins bound to something, and you're using actin and myosin together to create this contraction movement, as illustrated here, and to pull everything off. And remember, this is a motor protein moving, so you can imagine in this example, well, not imagine, that's what it's showing you. Here's actin, and here's your myosin bound to a surface, and so it's pulling the actin this way. All right? Any questions about that? All right. This is what that looks like. So before it plays, let me tell you this is a fish fibroblast cell. You have actin in red, and the myosin, the motor protein, is in green. Notice already how the red and green are so closely associated. This is the front of the cell. You're going to see things you've already heard about. Here's a filopodium. Here's a lamellipodia, right? So this is all the stuff you heard about. Here's all this actin that's being polymerized to move the cell this way. What I want you to watch is what happens back here. Myosin will work with actin to retract the cell, basically what I was just showing you. So the cell is going to want to move this way. It'll probably kind of, yep, it's growing in that direction. You see new actin, and then it pulls this backside right off. And it's because of the contraction that's happening with the myosin walking along the actin filaments. Okay, any questions about that? Yes?
So it just moves it in a straight
More or less, yes. Yeah. And there's so many of them that the overall direction of movement is going to depend on the sort of average force, but yes. Okay, so now we have about 20 minutes to go. And in preparation for your exam, I have this sort of summary slides that I had put together. They are simply meant as a guide about some of the topics we've covered. You may have noticed if you looked at it, it didn't include today's slides because, I'm presenting them today and it's pretty close, so I figured that would be fine. But it includes slides for everything that we've covered prior to today that are sort of the main slides that I think are important to learn. And I'm just kind of going through these in case you haven't looked at them yet, and you can pause them in lecture capture and look at them in detail. So you might recognize these things that we've covered, okay? So it's this series of roughly 15 slides. What I would like to do, I can go through some of these concepts in the next 20 minutes, but before I do that, I would really like to entertain any questions you might have, because I don't want to review the stuff you already know. I'd like to help you with the stuff you don't know. So if there are any questions about anything we've covered in the Unit 3, now's the time to ask. And if not, I'll start going through some of the stuff. Yes?
Should we really detail attention to free and tyrosine kinase complex? What is the major focus for tyrosine kinase?
So those types of questions, the should I pay attention to something, my answer is always very simple. If I taught it to you in class, it's on the exam. If I didn't, it's not. And if you go back and review my lecture captures, it's very clear what I'm emphasizing and what I'm not. You have bullet points of things to study. You just have to kind of pay attention to that. So, I'm happy, however, to answer any scientific questions about things you are confused about, concepts. You guys all understood it all super well, huh? All right. I don't think I'm that good of an instructor. Pardon me? Yes. Sure. Yeah, that's a common one that is complicated. So I think it's probably in these slides. If not, I'll pull up the original. Let's find that. Yeah. This, right? Or, let's see. Probably this one? Yeah, this slide? Yeah. So review of how GPCRs work as shown here. The background for this was you had a ligand that bound to this receptor, the 7-transmembrane G protein coupled receptor. Before that happens, there's no ligand here. Ligand is shown in red. This complex here is sitting here doing nothing. And that complex has 3 components. There are G proteins, alpha, beta, and gamma. The alpha and the gamma are shown here as tethered to the membrane, and the beta is sandwiched in between. When the alpha is bound to a GDP, it's off. The concept of a molecular switch, just like we talked about a few slides back with the row rack stuff, I think it was. Row, yeah. So you have this complex sitting here not doing anything. When a signal comes through, the receptor activates the complex. So it binds to and changes the shape of this complex, releasing the GDP and putting in its place GTP. So the receptor is acting as a guanine nucleotide exchange factor because it is stimulating the exchange of the GDP for GTP. All right, so the receptor is a GEF. Once that happens and the GTP binds to this complex, as soon as it binds in, instead of the GDP, the alpha separates away from the beta gamma. All right? So it's kind of a simultaneous thing. You have this as one complex, the GTP binds instead, it pops away from the beta gamma, and now you have your alpha with the GTP attached. The red dashes here indicate activity, so the removal causes shape changes that activates both the alpha subunit and the beta gamma subunit. Now, what the beta gamma subunit does is very context dependent. Sometimes it might be working on an entirely separate pathway, other times it might be sitting around not doing anything, it just depends. But the alpha subunit, to my knowledge anyway, is always activated and is taking then its own signal transduction pathway downstream. And it's going to do that until it hydrolyzes the GTP it's bound to be back to being a GDP. When you do that, you turn yourself off. And if you are hydrolyzing something, whether it's another protein or yourself, to go from a GTP to a GDP, that means you are a GTPase. You're an enzyme that acts on GTP to turn it into GDP. So the whole complex is turned on, the signaling is turned on by the ligand binding to the receptor, and it turns off on its own because its default state is to go back to being off. Any specific questions from you or anyone else on what I just said? Yes?
How then it activates second messenger again?
So let's see if I have a slide for that. I'm sure I do. No. Yeah, here we go. This is a good one to start with. So there's several examples of it, but we covered two major pathways in class. This is one of them that were downstream of GPCRs, where now that you have activity, so you have your activated G-alpha, it's going to go, remember the G-alpha, as shown here, is tethered to your membrane. So it can go bind, in this example, to something else that's also near the membrane, in this case adenylyl cyclase, a different enzyme, and activate that. Adenylyl cyclase, in turn, is going to take cyclic AMP, a substrate, and make cyclic AMP from ATP, the substrate. Flip that around in my head for a second. So once the cyclic AMP is made, that can now signal, and now the signaling has moved into the cytoplasm. So you're able to take a signal that came from outside the cell, and for a while you were restricted at this membrane, but now the signal is able to be sent into the cytoplasm through this process, because now the cyclic AMP will bind to and activate protein kinase A, pKa, and once that protein kinase A is active, it's then going to be able to activate other kinases and downstream and so on and so forth. So you really are taking a signal, it's like a relay system, like a baton in a relay race, where you're taking a signal from the outside of the cell, and because the signal itself is not going directly into the nucleus or something, you are instead using a relay system of sending the activation information through all these different components further downstream. Yes, that is one of the goals, absolutely, is amplification, because as you do this, every step of this is able to bind to many molecules. So what was one signal binding to one receptor can activate dozens or many more cyclases, can activate many more pKas, and all of a sudden you've got thousands of your downstream proteins the further down you go doing what you need to do, which helps it happen very quickly and efficiently. Yeah. Yes?
So CMP, doesn't it go through phosphodiesterase? Some of it goes through phosphodiesterase and other it goes through.
Yeah. So let me go to my. Surprised I didn't put this in here. I can pull it up in my actual lecture. There's a slide I really like for that. Here, let me pull it up. There we go. All right, so don't worry about the graying out right now because it's just from some point in the lecture. But this is what you're asking about. So we covered a few different pathways in class. We did not go into this one at all. But I told you just need to be aware of it. And we really didn't talk much about this one either. This is the main one we focused on for GPCRs. And these are ones I really told you about through receptor tyrosine kinases, which is not your current question. So we'll focus then on what you're asking about. So I just took you through in the previous slide G protein to adenylyl cyclase, cyclic AMP, pKa. Your question is, what's phosphodiesterase doing, I think? Is that kind of what you're asking? No? Oh.
Always go through phosphodiesterase, like when does it happen?
Right. So when is phosphodiesterase active? When is it doing something? So phosphodiesterase is one of these breaks in the pathway. We've talked a lot about, say, inhibition, the upside down T as a way to break something, where it's a direct interaction where let's say something else is moving the stool and I just want to inhibit it, I'll inhibit it directly. But instead of inhibiting the stool directly, in this case, what if I just turn the stool into something else? So that's what this enzyme is doing. Instead of inhibiting the cyclic AMP from doing anything, it just takes it as a substrate and turns it into AMP, adenosine monophosphate. Yeah, and that once you do that, you're decreasing the amount of this. And if you don't have enough of this, then you're not going to activate a lot of pKa, and it slows down or stops your process. So it's, if you go back to my analogy about rivers and dams and all that, so signaling is like a river flowing, and if you inhibit the flow, you're putting a dam in the water that stops the water from moving. I would view phosphodiesterase as digging a whole new tributary or sort of an outlet for floodwaters or something like that, where you're actually just taking this water flow, you're digging a new channel going in a different direction, and it diverts the water away because it's changing it into this AMP, and now you can't go the way you're supposed to go. Pardon me?
AMP does others.
AMP does other, well, so AMP, adenosine monophosphate, is the single phosphate form of ATP, ADP, right? And I don't know off the top of my head if AMP is directly used for anything or if it's just phosphorylated to become ADP, ATP. It probably does have some function. I don't know.
I think it's the most glycolysis already, because if we have a lot of AMP, that means we need to make a lot of ATP.
Okay, there you go. Right. So a lot of AMP can stimulate the proliferation of glycolysis because it's an indication to the cell that maybe it's running out of energy sources. So, yeah. Yes.
So before the signal that moves to proteins further down, it will always activate a kinase?
Say that again.
Will the signal always activate a kinase before going to the other proteins that you're specifically?
Oh, I see. Will the signal always activate a kinase before going to the protein? In this pathway, yes, because many of its components are kinases. A large percentage of cell signaling is happening through kinases and phosphatases. Not all of it, but a large percentage of it. So in this case, yes. The way to think about it is that your end effectors, effector molecules that we talked about that cause a response, they might be some other type of a protein, right? They might be something that's maybe it's directly, I don't know, maybe it's actin that's causing the cell to grow as your end effect. Maybe it's a transcription factor that's binding inside of your DNA to activate transcription. But if you go upstream of those things and you look at all the proteins that are causing you to get there, there are going to be a lot of kinases there, for sure. Any other questions? Yes?
The PDE you said is not membrane bound,
Correct.
but the adenosine is.
Yes. So the question was, is PDE not membrane bound, but adenyl cyclase is? And that's correct. Yeah. So if you look at this diagram, no. Oh, we're in the wrong. So if you look at this diagram here, so the adenyl cyclase is in the membrane and it's activated. The step at which the signal is able to go further into the cell is after that. And then the inhibition that has to happen with the phosphodiesterase is down here. So, well, it's actually here, basically at this step. So that would happen in the cytoplasm. Somewhere in all the slides, there's a couple of different diagrams that kind of show that. This is one example of it. Anything else? Yes? Basic questions are great. I have them all the time.
I know confused about the pathways, the arrows and stuff, the two arrows.
Sure.
And there, it's like, it's kind of positive. I really can't.
Okay, yeah, let's go over that. So this slide, right, probably is a good way to practice. Okay. All right, so I'll just kind of give you, I guess, the same overview very briefly that I did before. So when you're talking about a pathway, upstream means earlier, so A is upstream of B. Downstream means later, B is downstream of A. If you see an arrow like this, it means you're activating it. And an important point, that doesn't tell you if you're making more of it or just making it more active. You just know that you're activating it. It's a very broad term. And then upside down T's, which is what you're asking about, are inhibiting. So this example here, AKT acting on FKHR, what this means is that if you have more AKT that's doing its job, it's going to either decrease the amount or activity of FKHR. Now where people tend to get a little confused is when you've got a couple of inhibitions in a row or multiple inhibitions in a row. And if it helps, I think of it as -1 times -1 equals 1. It's the same general idea. In fact, it really is. So what's happening here is FKHR in this example here is inhibiting these receptor tyrosine kinases, right? So if I've got, ignore the AKT for a second, if I've just got a ton of FKHR being active, if it's active, what it's doing is inhibiting this, so you're going to have less activity of RTKs. If I now add a bunch of AKT to the system, that AKT is going to inhibit levels or activity of FKHR. If FKHR goes down, then this inhibition doesn't work as well. And if it can't do its job, you can imagine this going away. Now the RTKAs go back up because there's nothing stopping them from being there. So that's how the repression of a repression can lead to activation, because you're repressing here, you're repressing here. And so at the end of the day, if you're doing both of these things, you're going to end up activating this one because the repressor here is being repressed. Does that help? Kinda? No? What is what now? Oh, this lapatinib? No, it's describing, I think that's actually a drug. It's describing something that's going to inhibit the RTK. So it's a drug that's inhibiting receptor tyrosine kinases. If I had to guess, that probably means it's probably some sort of an antagonist or something that binds to it and stops the RTKs from doing their job. Yeah. Anything else? We have a few more minutes left. Yeah.
So the microtubules, we discussed that there was a cytoplasmic and axonal. Could we
Go over that? Sure. Cytoplasmic versus axonal microtubules. I think I do have some review slides for that right here. All right. Yeah. So we spent a lot of time talking about cytoplasmic microtubules, which are start off at a centrosome, which has centrioles in it, and a matrix with gamma tubulin, and that gamma tubulin, the slide I'm thinking of is not here, but you can imagine it. Gamma tubulin would be right there, and then you've got microtubules that are grown on it, and that the components are added to the plus end of the growing microtubule. In this diagram here, we're looking at dynamic instability. So as that microtubule is growing with GTP-bound tubulin being added, the bound GTP is indicated by the red dots, and you're growing in this direction. If you start to have hydrolysis, basically at some rate, the GTP is being converted back into GDP, and so the colors change as shown here. Now you no longer have as stable of a network of the microtubules, and that can more easily come apart. So if you lose eventually this GTP cap, over time you will, if you're not stabilizing it with something, then you end up with this situation, where now you no longer have your cap, and this end is very vulnerable to being dissociated. Inside there's enough stability that it generally stays together, but the ends are kind of available for something to tear it apart. And so now it'll start being torn apart, and you end up shrinking them back down. So that's cytoplasmic microtubules in a nutshell. They're involved in growing these highways that send things throughout the cell with the motor proteins and do a lot of wonderful things that I don't have time to go into complete detail right now. Axonal microtubules, on the other hand, work very differently. You have pairs of microtubules on the outside of this cilium, and you have a pair in the middle, which is called a 9+2 array, which is confusingly named because it's talking about 9 pairs and then 2 singlets here. And these microtubules, they're stable. They're not undergoing dynamic instability. They're there in the cilium or the flagellum on top of this basal body. And they have proteins attached, one of which is the motor protein dynein, and another one is the protein nexin. So when you have dynein being activated with an energy source, it can't go anywhere because it's in place, it's stuck between microtubules, so the energy of its movement is translated into trying to walk the microtubules past each other. That's what they would normally do, except that nexin binds them together. And that binding, I don't have it on here, but the binding causes then, instead of them moving like this, for them to bend. And when that happens really, really fast over again in a synchrony, can't all happen at the same time, but they do it in sort of a synchronous motion, you end up causing a bending and rotation of the cilia, and then it moves really fast and you get the motion. So you have some of the same components. You've got microtubules, but they're arranged differently. They're stable. They use dynein only. They don't use kinesin, which is used in cytoplasmic microtubules. And nexin is really important for the bending. It's kind of a brief summary of that. Okay, so class is over. I'll be upstairs until 2 p.m. So come on by and ask your questions. That's okay.
one centrosome? Or can a cell have multiple centrosomes? Or
Yeah, oops.